Thanks. It's a pleasure to be here. So just to get my conflicts of interest out of the way, I'm from a corn farm in Nebraska. I'm going to be talking about corn and biofuels, and so that's the corn farm. And I started a couple of companies in this sector, and I run an institute also in this area. So you all know this, but we get almost all of our fuels and almost all of the chemicals we use on a daily basis from petroleum, and that petroleum comes from the ground, and of course we pump it up and we refine it to make all the chemicals and fuels we use. And if you just look at a typical barrel of oil, roughly 85% of that is used to make fuels, things like diesel fuel, gasoline, and jet fuel, and the remainder of that goes into all of the chemicals we use. Now, the challenge, and most of you know this, is that our resources are dwindling. If you look at peak discoveries in the U.S., they happened in the early 70s and have been declining since, and the rest of the world is following suit. So we've had a lot of oil that's been produced, but those resources are declining, and the discoveries are expected to decline going into the rest of the century. Now, that's coming at the same time that energy demand is increasing. So in green is the OECD countries and their energy use, projected energy use over time, and you can see that the non-OECD countries are growing rather dramatically in their energy demand. Now, if we just take a few of these, in particular, U.S., shown in blue, of course, still has per capita very high energy demand, but China is going up dramatically. India is also going up dramatically, and these are countries that have half the world's population, and so we should expect to see that oil demand is going to go up dramatically. Now, the repercussion of that is that the price is projected to increase, could increase rather dramatically. This is some of the worst case scenarios. I don't really believe this low price scenario, but prices are probably going to be somewhere between the reference point and this high price, which means that fuel costs will increase for all of us. Now, at the same time that we have this increased fuel use, we're also putting a lot of carbon dioxide in the atmosphere, and that's resulted in an increase in temperatures. And this is, of course, projected to increase rather dramatically over the next century. And you can see that a lot of the carbon dioxide is predicted to come from non-OECD countries as their economies grow. Now, one possibility to get around this is to make our fuels from a renewable resource. That could be from corn in the U.S., that could be from sugarcane in Brazil, cellulosic materials, or even grains, oils that come from grains. Now, the nice aspect of this process is that sunlight and carbon dioxide can be fixed to produce the biomass. In this case, I'm showing corn here. That corn is then grown on farms, collected, refined in a biorefinery into the fuels that we might use in our automobiles. And because you burn those fuels in the automobile, you create carbon dioxide, but you have the potential of having fuels that are carbon neutral. Now, if we just look at ethanol as a fuel, as one of the fuels that we might use, you can see that world production of ethanol has increased rather dramatically. So this is all due primarily to fuel use. There's been, good news is there's been little increase in beverage consumption of ethanol or industrial use. It's been primarily for use as a fuel. And if you just look at over the last 10 to 12 years, it's increased dramatically. And a lot of that increase is in Brazil and the United States. And if you just look at the amount produced annually in Brazil and the U.S., it's on the order of 5 billion gallons per year. So that would be on the order of 18 to 20 million liters annually. In Brazil, from sugarcane, in the U.S., from corn, and in other parts of the world, in Europe, for instance, sugar beets, wheat, and sorghum. So a variety of sources of biomass to produce the ethanol. Now, there are a number of challenges in this production of fuels from things like corn. And the first is just the corn itself. Corn, as you may know, requires a lot of water. It requires a lot of fertilizer. And an additional challenge is that it's used to feed cattle primarily in the U.S., and of course those cattle are beef. And so there's this concern over us trading food and fuel. If we just look at the energy it takes to produce corn, it's pretty substantial. And that energy is tied up primarily in the production of nitrogen-based fertilizers. So just to give you a rough number, roughly 1% of the world's energy goes into producing nitrogen-based fertilizers that are used to fertilize corn in the U.S. that goes to produce beef. That's a third of the energy used in agriculture. And if we could find a solution to make corn and other crops fix their own nitrogen, it would be a huge savings in terms of energy and in terms of the amount of carbon dioxide we put into the atmosphere. Other uses of energy in production of corn are, of course, the fuel and all the other chemicals that are put on corn. Now, that in and of itself makes corn very inefficient to use as a source of energy. So I've plotted here four different crops: corn, sugar beets, switchgrass, and sugarcane, and the amount of energy that can be expected to be produced from those when you're producing something like ethanol. So corn produces roughly 34% more energy than it takes to produce it. So it's just barely energy productive. In fact, there have been some calculations that have shown it's even been negative. Now, if we compare that to something like sugarcane in Brazil, sugarcane produces roughly 7 to 8 times more energy than it takes to produce the sugarcane itself. Now, in the U.S., we're looking at other potential sources for producing those fuels, such as switchgrass. And this has a number of advantages. One advantage in particular is that if you produce ethanol from it, you could get up to 4 times more energy out than you put into growing the crop. And so this would substitute for the starch that you get in corn, but yet would be this cellulosic biomass. Now, there was a study done in 2005, and it was updated in 2011 to project how much biomass we might have available in the U.S. And I apologize that these numbers are rather U.S.-centric, but they're the numbers I know the best. And it was projected that by 2030, we could have somewhere on the order of 1.3 billion to 1.5 billion tons of biomass available annually that could be turned into fuels. Now, as you'll see, it's primarily in these green bars, which are dedicated energy crops. It could also be in waste trees that have fallen down into forests that are managed. For instance, it could also be parts of the corn, like corn stover, which is everything but the kernel of corn, that could be used to produce that ethanol. But on the order of 1.3 billion tons annually could be turned into fuels. Now, to give you some perspective of how much fuel you might produce from that 1.3 billion tons of biomass, I want to compare it to the energy used by the U.S. in 2007, specifically barrels of oil. So we used about 7 billion barrels of oil in 2010. Roughly half of that was imported, and half of that was domestically produced. Now, if you just take that 1.3 billion tons of biomass and burn that, you'll get roughly the energy that you get from the domestic production of oil in 2010. So that's a substantial resource in terms of the energy. But not all of this biomass can be turned into fuels. We project that about somewhere between half to 2/3 of that could be turned into transportation fuels. So we might get on the order of 2.3 billion barrels of oil equivalents from that 1.3 billion tons of biomass. That'd be substantial. So that's on the order of a third of the energy used in 2010. That would be a substantial change in the amount of carbon dioxide put into the atmosphere and a substantial change in the distribution of money in the world. Now, let's go back to this carbon, putting carbon into the atmosphere. I've already talked to you about the fact that the amount of carbon we've been putting into the atmosphere has grown dramatically over the last century. If you were to take a crop like switchgrass, which is a grass that grows natively in much of the U.S. Midwest, and a grass that's been touted as a potential energy crop, if you plant that and harvest it each year and turn it into ethanol or another biofuel, I'm showing here what that would do for the carbon footprint of those fuels. So these are greenhouse gas emissions that would be displaced by producing fuels from switchgrass. And what you see here is that it's even greater than 100%. You might ask, why is that? Because we're growing the switchgrass, we're harvesting it, we're extracting the sugars from it, turning it into fuel, and burning that fuel, and that carbon dioxide is going back into the atmosphere. So how come you could have greater than 100% carbon removal? Well, it turns out that switchgrass is very effective at fixing carbon and putting it into the soil. And if you saw Paul Alivasatos' talk yesterday, he probably told you about all the carbon that's bound up in the soil. Switchgrass turns out to be an excellent fixer and store of carbon in the soil. In fact, there are microbes that live around the switchgrass that store that carbon in the soil. And so it's projected that we could even increase the amount of carbon put into the soil by growing switchgrass and turning it into fuel. Now, the challenge, though, is turning that biomass into fuels, because biomass is really complicated. So if we just look at the structure of biomass, it's shown here. This is a micrograph of a plant wall. And after the plant cells die, which were in these cavities, all that's left over is the plant cell wall. And this is where all the biomass is that we want to turn into fuels. Now, that biomass in and of itself is really complicated. It's made of primarily three components. Lignin is an aromatic polymer, shown here in red. And then there are two polymers that are sugars, hemicellulose, which are sugars that are not the best sugars, and cellulose, which are very good sugars. Now, if we just look at a typical plant, doesn't matter what the plant is, whether it's trees like Douglas fir or poplar trees, or things like cornstover or grasses, they're roughly half to two-thirds sugars. So let me say that again. Plants are roughly two-thirds sugar. So that's a huge resource that we could use and turn into fuel. Now, in order to extract the sugars from that plant, we have to run it through two processes. We have to use a pretreatment process, which extracts the lignin. This lignin on plants gives the plant both its structure. It also protects the sugar so that all the creatures in the environment, like the dung beetles, like fungi, won't chew up the plant and get access to the sugars. So we have to extract that lignin. We use pretreatment processes that tend to be very rigorous, high pressures, high temperatures, low or high pH. And this gives us a sugar stream that's not completely pure, but that's still bound up in these polymers. Now, the challenge with a polymer of sugar like cellulose is that it's still very tough. And let me give you an example. This shirt is cotton, and cotton is pretty much 100% cellulose. And yet when I put this shirt in the laundry, in water, it doesn't dissolve. And I can do that over and over again, and yet it's 100% sugar. That tells you how strong that sugar is in this polymer. So we need enzymes. These enzymes tend to come from microbes in the environment that degrade cellulose. Those enzymes break up the cellulose into the individual sugars, and then we feed those sugars to microbes like yeast that turn it into fuels like ethanol. Now I'm going to talk about some of the challenges today and how some of the solutions to address those challenges. So one of the challenges is we don't have as much sugar in those plants as we'd like. We'd like to have even more sugar, 75%, 80% sugar if we could. We'd like to have as little lignin as possible because we can't make anything from that. There's an old saying, you can make everything from lignin except money. Another challenge is that we'd like to have better pretreatment processes to extract the lignin away from the cellulose and hemicellulose. The better the pretreatment process, the easier it is going to be to turn those into sugars. During those pretreatment processes, because they tend to involve high temperatures, high pressures, we generate a lot of inhibitors, and those inhibitors then inhibit the enzymes that break down the polymers into the sugars and inhibit the microbes that produce the fuels from those sugars. And finally, or third maybe, the enzymes are very expensive that we add to break down the cellulose and hemicellulose into sugars. They add anywhere to around $1 to every gallon of fuel that's produced from cellulosic biomass. So $1 to every gallon of fuel. That's a huge cost. And finally, we'd like microbes that would produce fuels for all of our modes of transportation. Ethanol is a fine oxygenate for gasoline, for automobiles, but it can't be used in diesel engines and it can't be used at all in jet engines. So we'd like to have fuels that are identical to the petroleum-based fuels that we get from oil right now. So the institute that I work at actually works on all aspects of turning sunlight into advanced transportation fuels. And I'm going to talk about a few of the discoveries and solutions we have to address some of these challenges. So the first challenge I'd like to talk to you about is this problem with the lignin and how it binds the cellulose and hemicellulose in the plant. We'd like to reduce this to the extent possible. Now, there are two ways around this. One could develop modified plants that would have less lignin in them, so that when you strip it away, you get a cleaner cellulose and hemicellulose. Another possibility is that you could develop a better pretreatment method that would extract the lignin from the cellulose and hemicellulose. So I want to talk about each of these, and we're first going to talk about the plant side of this. So this is artist rendition of what lignin looks like. It's a polymer that's very diverse in its chemical structures. It's highly polymerized, and as a result, it's very difficult to break lignin down and remove it from plant biomass. Now, researchers at JBay have found ways to incorporate stops in the lignin so that you get shorter pieces of lignin. You don't necessarily have reduced amounts of lignin in the biomass, but it's shorter, and this allows it to be extracted easier. Now, the way we do this is we incorporate enzymes into the plant that produce chemicals that are incorporated into the lignin and stop the polymerization, so it makes lignin that's shorter than what you'd normally find in the plant. Now, here's the data. The first thing we do when we do these engineering of the plants, we first look to see if the plant will grow, because a lot of the changes we make in a plant end up killing the plant, and so it won't grow. And you can see here that these plants with the modified lignin look the same as the wild-type plant that we started with that has the normal lignin. Now, this is difficult to see, but these are data that basically say we have shorter pieces of lignin, still the same amount of lignin, but much shorter lignin. And then when we subject the biomass, these plants, to pretreatment to remove the lignin, we get substantial increases in the amount of sugar released, increases of 60% or 30%, depending on the pretreatment method that you use. That increase in the sugar release means that there's an increase in the amount of fuels produced from the plants. Now, often, when we make these changes to the lignin, we get plants that look like this. Now, it might have less lignin in it, but this obviously isn't going to be a good producer of biomass because it competes with a normal plant that grows well. So, when we got some of these results, researchers at JBay said, well, maybe we can replace that lignin with cellulose in parts of the plant. And in an ideal world, what we'd do is we'd have wild-type amounts of lignin in the stems. These help to keep the plant upright, but we'd fill the leaves of the plant with more cellulose to make up for the lignin. Now, the way you do this is you engineer transcription factors. Now, in you and me, transcription factors are responsible for making sure that fingers end up on our hands and toes end up on our feet. They control massive numbers of genes and regulate those genes in different parts of humans or in different parts of plants. And so Dominique Loki, one of the researchers at JBay, found a way to change the transcription factors so that we could add more cellulose in the leaves of the plants and yet keep the lignin in the stems of the plant. And so here's the original plant where we had reduced the amount of lignin. Here's the modified plant. You can see it's got a different color, but it's growing just as well as the wild type plant. This has got more cellulose in the leaves. And just to show you the evidence that it has more cellulose in the leaves, here's a control plant. Here's before we started to do the engineering. You can see these big holes in the plant cells. Here's the engineered plant. You can see now that these holes are completely filled up with cellulose. We get a substantial increase in the amount of sugar stored in this plant, and that means that from a given amount of plant biomass, we'll have more fuels produced from it. On the order of 30 to 50% more fuels, depending on the pretreatment method that you use. Now, I mentioned that there's another avenue for getting cleaner cellulose and hemicellulose, and that's to use a better pretreatment method. Traditional pretreatment methods, as I indicated, usually involve high temperatures, high pH, and this means that you have to build a biorefinery with a very expensive stainless steel to withstand these extremes in temperature, pH, and pressure. And so researchers at JBay have been working on a technique, a method called ionic liquids. And these, that's shown right here, these are ionic liquids. They're like table salt, except that they're liquids at room temperature and pressure. You mix them with biomass. This is ground up switchgrass. You add a little heat and you stir, and it becomes like molasses. So it's a dark liquid. Then when you add water back, you get from it almost pure cellulose and hemicellulose coming out of it. And this just shows a micrograph of what happens to the plant cell wall. This is the starting plant cell wall. After a few minutes with ionic liquids, they swell, and you can see that the cellulose is now starting to come out of the lignin cellulose structure. After about 3 hours, you don't see any structure left. When you add water back to it, the cellulose precipitates out, and this is really essentially pure cellulose and hemicellulose that's coming out of the plants. Now, to just show you that we're getting better material out of that, the next step is to take that purified cellulose and hemicellulose and subject it to enzymes to look at how quickly they can degrade it into sugars. Here's the traditional pretreatment method involving dilute acids. It takes about 70, 80 hours to depolymerize the sugars into individual sugars. Here's the ionic liquid pretreated biomass. It takes about 7 hours to do the same amount of depolymerizing the cellulose and hemicellulose into sugars. This lower time means a lower cost for the ultimate cost of the fuels. And it doesn't matter what biomass you use. You can use eucalyptus trees, you can use corn stover, switchgrass, and this pretreatment method seems to work equally well regardless of the amount of biomass or the type of biomass you use. Now I want to talk just briefly about another challenge we're trying to address, and that is these inhibitors that result from the pretreatment methods that poison the enzymes and the microbes. And these inhibitors come from the biomass itself. Well, you can't see the cellulose and hemicellulose up here, but basically these are decorations, chemical moieties that are put onto the hemicellulose. And when we process the biomass, these are released from the hemicellulose and result poisoning the microbes that produce the fuel. We don't know what they're doing on there, but we'd like to remove them. So researchers at JBay went in to look for the enzymes that are responsible for adding these to the hemicellulose. They found the enzymes, they deleted some of those genes, and they got a reduction in the amount of acetate in the stems, which means less acetate or less inhibitor that ends up in the sugar that produces the fuel. And when you get that reduced amount of acetate, you actually increase the amount of sugar released rather substantially. So you can see here we've gone from a sugar recovery of about 25% to 100% in xylose, so a substantial threefold increase in the amount of sugar released. And we think that what's happening is that we're breaking the bonds in this biomass and allowing the sugars to come out more readily from the biomass because they're not linked or cross-linked to the lignin. Now, when you're working with plants, you can never predict what the result is going to be. There's so much unknown in engineering plants. Often what happens is that those plants don't grow or they become susceptible to pathogens. And here's an example where something very positive came out of it. So one of the tests we do is we subject the plants, the wild type and the engineered plants, to a pathogen. And this pathogen, over time, makes these big lesions on the plant leaves. And this is a huge problem. What happened actually very positively in these engineered plants with reduced acetate is when we subjected them to the same pathogens, those pathogens weren't nearly so effective at destroying the plant. So this is actually something that happened. We didn't expect it, but very positive. Now, for the rest of my time, I want to spend talking about this last aspect of the problem, and that is how do we engineer microbes to produce advanced fuels that we can use in all of our modes of transportation. Now, our process for doing this is to engineer the chemistry that goes on inside the cell and get those cells to produce fuels that are identical to the fuels we now get from petroleum. And I want to talk about that science. Now, the first thing you have to think about if you're going to engineer a microbe to produce a new fuel, a fuel that hasn't been produced, is all the factors in our existing fuels. So you have to think about what kind of engine you're going to use it in, whether it's a diesel engine, a gasoline engine, or a jet engine. It's got to have high energy content because this is how far people can drive on a given gallon or liter of that fuel. It has to have a good combustion quality so that your engine sounds the same as you're driving down the road on it, whether it's a renewable fuel or a petroleum-based fuel. It can't be cloudy because those will plug the filters in the fuel for the fuel. It has to be volatile, just the right volatility. It's got to be stable because we want to store it, we want to ship it through pipelines, we want to put it into our tanks and store it for periods of time. It can't be smelly. People don't like smelly things. I can guarantee that our tanks are going to leak, and so it can't be toxic when it gets into the environment. We don't want it miscible with water. This is one of the challenges with ethanol. We can't pipe it because it picks up water as it's being piped, and this means that ethanol has to be shipped by rail car or by train car or truck. And, of course, it can't be 4,000 krona a liter. So here are some of the hydrocarbons that we've been working on at JBay, and we picked these hydrocarbons because they match both the biochemistry that we think we can do inside a cell and also the types of fuels that we now get from petroleum. And I just want to outline some of the work we're doing on a few of these. So the first fuels I want to talk about are these fuels that have these long alkane chains. So these are very good as diesel and jet fuels. In fact, our diesels and jet fuels have a lot of these in them, particularly in the summertime in diesel fuel. Now, one source for these fuels is plant oils. Things like sunflower oil, canola oil, are great sources for biodiesel, but there are a couple of challenges. One is these aren't very productive per acre. You get on the order of hundreds of gallons, maybe 200 gallons of biodiesel per acre. So that would be, about 1,400 liters per hectare. So limited amounts of biodiesel from these sources. The other challenge is that you have to take the oils you get from these and esterify them with an alcohol before you can use them in your diesel car. So we said we can do better than this. We ought to be able to get microbes that will produce a diesel, take in a sugar source, produce both the ethanol and the fatty acid, and do the esterification inside the cell. So you eliminate a chemical step, and you're able to use sugar, which we can produce lots of on a per acre basis. And so when you engineer some organisms like E. coli or yeast to produce these fuels, the interesting thing is that they actually secrete the fuels. So the fuels come out of the cells and float in the medium. And so here's E. coli that's been engineered to produce this diesel, and these are the blebs of diesel that actually have been secreted outside E. coli. This is a culture tube here, and floating at the top here is the biodiesel. And because we've tailored the chemistry inside the cell, you don't have to do further processing of this fuel. So there are companies now that have been launched based on this technology and that are using it. They grow the microbes up in huge tanks with stirrers. They turn off the stirring and the fuel essentially floats to the top. They run it through a very simple centrifugation process. It's basically a cream separator that we use to separate cream from milk as it comes out of the cow. Use the same process to now separate the fuel. And because we've tailored the chemistry inside the cell, the fuel is ready to use without further processing. Now, one of the challenges with diesels is that molecules like this will freeze up in the winter, or if you're at 35,000 feet in the atmosphere. And so you need branches in the long chains to break up the fuels from lining up. In fact, if you have all of this kind of molecule in your diesel tank in the winter here in Sweden, it'll turn into wax in your tank. So you need to install these branches inside on the molecule. And one way to do that is using a chemistry that's naturally occurring in many plants and microbes and even in humans based on isoprenoid chemistry. Now, we didn't get started on this chemistry producing fuels. We actually started our work in this area engineering microbes to produce this molecule, which is called artemisinin. It's a hydrocarbon. It's about the molecular weight of diesel fuel, but it happens to be the best drug for treating malaria. And through work funded by the Bill and Melinda Gates Foundation, we engineered a yeast to produce large quantities of this molecule, and that's actually going into production this year. The production facility will be in Italy, and they'll be producing 100 million treatments of this drug on an annual basis for malaria sufferers. We had engineered a microbe to produce this hydrocarbon, and it became very easy then to just change the chemistry inside that cell to produce molecules that would be good gasoline substitutes, would be good jet fuel substitutes, and diesel substitutes. And one of these molecules, bisabolane, happens to be an excellent diesel substitute. In fact, it's got properties that are nearly identical to number 2 diesel, which is the diesel that's used most widely throughout the world. It's got a cetane number, which is the number that's used to rate diesel in terms of its quality. That's almost identical to number 2 diesel. It's got a very low freeze and cloud point, which make it important for use in the winter under cold weather conditions, and it's got a high density. So very good potential diesel molecule. We engineered a microbe to produce bisabolene, and then we can use a simple hydrogenation process to remove the double bonds and get bisabolane as a diesel fuel. Now, one of the challenges in producing these fuels is that they're often toxic to the producer microbes. This molecule is a precursor to jet fuel and makes actually an excellent jet fuel. It also makes a great disinfectant. So I don't know if you have this product here, but in the US we have a disinfectant called Pine Sol, and it's basically an extract from pine trees. You spray it on your kitchen counter, and it kills all the microbes. Well, about 30 years ago, a scientist in the Boston area had discovered that when microbes are subjected to things like pinene, they turn on genes that produce pumps, and those pumps pump the pinene outside the cell. And it's very effective at making these cells resistant to pinene. It also makes the cells resistant to things like antibiotics, and this is one of the greatest sources for antibiotic resistance in microbes. Well, when Mary Dunlop came to J-Bay, she said, we ought to be able to find pumps that would make E. coli and yeast that we've engineered to produce this fuel resistant so that they can produce more of the fuel. So she actually cloned out pumps from several different organisms, put them into E. coli, and found pumps that would work very effectively at pumping this fuel outside the cell and allowing the cells to grow and produce the fuel. Now, the last issue I want to address today that we've been working on through synthetic biology is reducing the cost of the enzymes to depolymerize cellulose and hemicellulose into sugars. Now, one of the ways you could do this is to engineer the fuel-producing microbe to also produce enzymes so that you wouldn't have to add enzymes separately to degrade the cellulose and hemicellulose into sugars. The cost then would be reduced because that microbe would consume some of the sugars and produce the enzymes as well as producing the fuels. So Greg Bokinski, a postdoc in my laboratory, engineered E. coli with both 3 different biofuel production pathways, as well as the enzymes that allow us to take in ionic liquid treated eucalyptus or switchgrass and turn those into sugars, and then the cell would consume the sugars and produce the fuels directly. Now, I've talked almost exclusively about producing transportation fuels, and our whole goal has been to produce transportation fuels, biofuels that are identical to the fuels we now get from petroleum. But we also get a huge number of chemicals from petroleum. And I took a very long list and pared it down here so that you could just see some of the names of all the chemicals we get. You know, if you look around you, the seat coverings on your seats are made from petroleum. The carpet on the floor is made from petroleum. The paint on the walls and on the ceiling is made from petroleum. The finish on this floor is made from petroleum. There's almost nothing that we touch that doesn't have some root in petroleum. If we can produce fuels using engineered microbes, fuels that are nearly identical to the petroleum, to the fuels we now get from petroleum, there's no reason why we can't produce nearly all of these chemicals from sugars rather than from petroleum. And that's going to be the challenge, I think, going into the next decade and beyond, is how do we produce everything else that we get from petroleum, but now from sugar. These are some of the people at JBay. I've highlighted their work. I'd like to thank them for their efforts. I'd like to thank the U.S. Department of Energy for funding, and I'd like to thank the organizers for inviting me and you for your time and attention. I'd be happy to take any questions.
If you ferment carbohydrates and make ethanol, what is the yield in terms of energy in that process?
Yeah, so you end up expending quite a bit of energy because you're getting something that's you're getting ethanol out of the process. You've also got to grow the microbe. The fuel, it turns out, the yields are pretty good. If you just look at where we are in terms of theoretical yields, it's on the order of 90% of theoretical yield if you take a typical fermentation that's occurring in Brazil, for instance. Over the last, say, 3 decades, they've gotten the production process to be pretty good. They're getting very high yields of ethanol from it. Ethanol produced from sugarcane in Brazil right now is on the order of 90 cents a gallon. So the main reason is because they're starting with very cheap sugars, but the yields are extremely high.
So you don't lose too much during distillation, isolation, purification of the ethanol then?
Well, so it's a great question, and it's a complicated question and answer because if you look at, let's just take sugarcane in Brazil, and I think it's a great model for producing fuels worldwide. They extract the sugar from the cane. That sugar then is fermented. You get the ethanol out of it. The rest of the cane plant, the bagasse, they produce a lot of. They don't convert that into fuel, so what they do is they burn that. You get heat out of it. They get more than enough heat to distill the ethanol from the fermentation broth. The rest of the heat actually goes into making electricity. From the fermentation broth, they take that vinasse, they call it, and they truck it back out to the cane fields, and they fertilize the cane fields with it. So it's a completely renewable process. Does that answer your question?
Yeah, thanks a lot.
So it's somebody behind you there. You can send a microphone.
So I have a question about the lignin, because I wonder why it's so useless. Isn't there anything you can do with it, or see it as a possibility to create something, or try to research about stuff?
It's a great question, and it's a really important question. So there are researchers now that are looking at making carbon fibers from lignin. One could imagine that as we get better and better at engineering plants, that we could have dedicated energy crops that have lignin in them that maybe would be the perfect carbon fiber that you could extract very easily, that wouldn't be so tied to the cellulose and hemicellulose, so we could get the cellulose and hemicellulose out of the plant. One could imagine that if we could break up that lignin into its individual monomers, that we could then use those aromatic monomers for all the applications of aromatics we use now. So I think there's a lot that can be gotten from lignin in the future. It's a matter of engineering a plant to release it in the right way.
Since you can engineer organisms to use the energy in sugar to produce fuels, wouldn't it be possible to engineer other organisms to use that energy to produce electricity? I mean, much like an electric eel can produce electricity.
Yeah, certainly there's some possibilities. There's a thought that organisms pass electrons back and forth to each other on wires, essentially, and it's still rather controversial whether that exists or not, but there's certainly a possibility. There's also a possibility that in fact, the U.S. government is funding projects on this where microbes are attached to an electrode. You collect the electricity and pump it into the microbe, and then say you'd have a source of carbon dioxide, so that they could use that then to fix the carbon dioxide and produce fuel out of it. So it's just the opposite of what you were suggesting. And on paper, it looks to be a very efficient process. Whether it will actually end up being very efficient, time will tell.
Well, you've talked about developing fuels that are more efficient.
Yes.
I assume that engines also develop and become more efficient. Don't you risk being a bit behind developing fuels for the engines as they are right now, and then they develop and your fuels are outdated?
Right.
How do you handle that?
Yeah, it's a great question. So if you look at the automobile, the history of the automobile, and look at implications for the future, you see that it takes about 15 years to turn over a typical automobile fleet. So some people keep their cars for 4 years, some people keep their cars for 20 years. So on average, they usually assume that an automobile fleet turns over in 15 years. So that's essentially the time frame we need to think about. Now, if you just look at changes in engines and just think about when we had the last major change of an engine, it's been a long, long time. Engines become more and more efficient, but they generally are still built around the fuels, diesel, gasoline, and jet fuel. And the changes in jet engines are even longer than that. So there is research on a new type of engine. It's a hybrid between a gasoline and diesel fuel engine. It runs much more efficiently than either diesel or gasoline engines. We're actually working with the scientists who are working on that engine. We're tailor-making the fuel producing microbe while they're working on the engine. And so I think as we go forward, there's these possibilities of the biology in sync with the engine
As a researcher in biofuels, have you heard complaints that our strive to streamline the energy production reduces the biodiversity by only growing certain types of plants?
Yes. So, I think sustainability is going to be a really important issue going forward. Frankly, we should have thought of sustainability long ago because there are many things that we've done for food and for other types of crops that have been harmful to the environment. But I think, given the size of the energy problem and what's likely to be, large amount of land devoted to producing energy, we have to think about diversity and sustainability. It's a huge issue.
All right, there's a question from the internet recently received regarding the biomass source after I found it. It's from Melvin L. from Singapore. Regarding the biomass source, after I found from the lecture that transcription factors control amount of lignin and cellulose in the plant, I realized that leaves contain more cellulose than lignin. So based on this factor, will dry leaves be considered to be a good source of biofuel or bioethanol, as there are lots of dry leaves which have been produced around the world?
Dry leaves.
Dry leaves, yes.
Yes. So one can collect biomass from any number of sources. And I think if we look at the size of the energy problem and how much land it's going to take, we're going to need very diverse sources of plants for producing these fuels. And so we'll have sorry, I'm stumbling on this answer, and we're going to have different sources of biomass depending on where you are in the world. So, for instance, here you might have trees as being the primary energy source. In places like the U.S. Midwest, it might be tall grasses. In places like Brazil, it's going to continue to be sugarcane. So, as it turns out, the amount of lignin in plant material is pretty much is very similar, regardless of what part of the plant it is or which plant it is. I mean, it varies a little bit, but if you saw that slide I put up with different amounts of lignin, cellulose, and hemicellulose, it didn't vary that much, regardless of the plant. So I think being able to engineer plants to be dedicated energy crops where they have less lignin is going to be an important aspect of the work going forward.
All right, so let's thank Professor Keasling once again.